Welcome to Chapter 1. This is covering Section 1.3. The best way to follow along with this video is to have your textbook out, be ready to pause the video often, complete problems on your own, and take notes for important vocabulary and terms. The key concept of this is all about collecting sample data. So the method used to collect the sample data actually influences the quality. And if you use a bad method or a method that collects inaccurate or incomplete data, then the data collected is useless. And we'll talk about simple random samples as well. We've got a lot of important vocabulary and terms, so please be ready to take notes. In general, the gold standard is randomization with placebo treatment groups, and it's sometimes called the gold standard because it's so effective. And maybe you've heard of the placebo effect in your life. You thought you took some medicine, you started to feel better, and then you realized you actually didn't take any medicine. So using a placebo can be an effective way to collect data, and we'll talk about that as well. So the basics of collecting data: we can have an observational study or an experiment. An observational study measures characteristics without attempting to modify, whereas an experiment is going to apply some sort of treatment. In general, experiments are often better than observational studies. So let's take a look at this experiment here. In 1954, an experiment was designed to test the effectiveness of a certain vaccine in preventing polio. So they selected 400,000 children at random. They assigned them to two groups, one group getting the treatment, the other given the placebo. And then children were assigned to the treatment or placebo group through a random selection. And then they found the results the following. And it looked like the vaccine might actually work. So go ahead and give this a try and press play when you're ready to see the answer. So there are a couple different ways to consider data in general. So let's think about data on ice cream sales and drownings. And these are one of my favorite examples when it comes to correlation against causation, which we talked about in a previous example, is two things may be increasing or decreasing or have some sort of relationship, but one doesn't necessarily cause the other. So we talked about ice cream sales and drownings. Let's see. Observational studies. So what that would mean is we would observe the past data and we would see that ice cream causes drowning because based on the data, there was an increase in ice cream sales and an increase in drowning. Well, what's going on here is that there's a lurking variable of temperature, and the failure to see that is temperature increases, people are going to buy more ice cream, and people are also going to be more likely to swim, and the more people swimming, the more likely there's drownings. So that observational study doesn't really help us consider the data on ice cream sales and drowning. So let's look at an experiment. If we were to conduct an experiment with one group treated with ice cream while another group gets no ice cream, we would see that the rate of drowning victims is about the same. So again, we would give one group ice cream, one group no ice cream, and see how many people drown, which is horrible but silly for this example. We would see that the rate of drowning really has no impact based on the ice cream. So we can see that an experiment in this particular case is better than an observational study. So observational studies can happen in the past, retrospective, one point in time, cross-sectional, or forward in time, prospective. So those are the three types of observational studies. There's a lot more to discuss when it comes to experiments. So the design of experiments should include the following: replication, blinding, and randomization. When we talk about replication, it's the repetition of an experiment on more than one individual. So it's not really enough to say that this worked on one person or one subject, but really needs to be able to be replicated. Good use of replication requires sample sizes that are large enough so that we can see the effects of treatments. If we're only looking at 5 people and we want to make an assumption about the entire population, that's really not going to be replicated enough for us to say for sure. We've got blinding and then double blinding. So blinding is a technique in which the subject doesn't know whether he or she is receiving a treatment or a placebo. And it's a way to get around that placebo effect, where we don't want people to think that they're doing better necessarily. We want everyone to kind of not know what's going on and see what's actually happening without that placebo effect. A double-blind experiment is where the subject doesn't know and the person running the experiment doesn't know, because there could be some inadvertent bias or things that can happen if the experimenter knows who has the treatment and who doesn't. And randomization. Randomization is used when subjects are assigned to different groups through the process of random selection, and we saw that in the vaccine example. The logic is to use chance as a way to create two groups that are similar. We're going to focus on 5 different sampling methods, and we're going to go into them a little bit more, but we've got simple random, systematic, convenience, stratified, and cluster. And in general, simple random is the best, and convenience is the worst. So you can pause and read a little bit about simple random. Systematic is every case element. So it doesn't always have to be TH. It could be every first person, every first female that walks in the door, every third, every fifth. And so we're looking at a certain number each time. So that's a way to get random, but also be systematic in how we're choosing random. Convenience, if you think about maybe asking the first 10 people that walk by you, standing outside of a grocery store and asking the first 10 people you see, or maybe on Facebook posting a question and whoever answers. Stratified is subgroups. So finding something that makes people similar or different and separating them to see what happens in the different strata or groups. The difference between stratified and cluster is location. I always like to bring students' attention to like the L in cluster for location. So we're really looking at location when we talk about cluster sampling. Maybe by zip codes, maybe by blocks, maybe by states. So go ahead and pause this and identify which type of sampling is used for each of these and press play when you're ready to see the answers. Multistage sampling is where we use a combination of the different sampling methods. So here's an example of a U.S. government survey. And so at each different level here, they're combining random, stratified, cluster at different stages. The end result is a very complicated sampling design, but it may be more practical or less expensive and faster than a simple random sample, which can actually be quite difficult. Confounding occurs in an experiment when the experimenter is not able to distinguish between the effects of different factors. So trying to plan the experiment so that confounding does not occur is really important. So if we think back to the ice cream and drownings, there's something we don't understand that's going on here. We're not able to distinguish what is having an impact on another. When we think about bad experimental design, here's an example. If we were to treat all women subjects and give the men a placebo, the problem is we don't know if the effects are due to the sex of the individual or to the treatment. So that is a bad experimental design to say, okay, all men over here, all women, or if we separated by age, if we said over 30 in one group and under 30 in another, we wouldn't know if age or the treatment really had the effect. If we had a completely randomized experimental design, we would assign subjects to different treatment groups through a process of random selection. And again, that was talked about in the vaccine example. So it's totally random, and so we don't know who's getting the treatment and who's getting the placebo. We could do a randomized block design. So a block is a group of subjects that are similar, but blocks differ in ways that might affect the outcome of the experiment. So we would form a block of women and a block of men. And then within each block, we would randomly select subjects to be treated. This way, we know we're getting a sample of women and a sample of men, rather than totally random, which may not turn out the way we wanted. And matched pairs design is where we compare two treatment groups by using subjects matched in pairs that are somehow related or have similar characteristics. So we're looking for the before and after. Rigorously controlled design was where we carefully assign subjects to different treatment groups so that those given each treatment are similar in ways that are important to the experiment. So the main focus here is we're just trying to control the effects of variables. We don't mean like X, Y, Z necessarily. We mean confounding variables that happen in the real world that would make an impact when we're trying to determine the impact of one thing and other things are creating the impact instead. There are other things we need to be aware of, like sampling errors. So no matter how well you plan and execute that stratified collection process or systematic, there's likely to be some error. So here are 3 sampling errors. The first is random sampling error, just sampling error. And it occurs when the sample has been selected with a random method, but there's a discrepancy between a sample result and the true population result. Non-sampling error is the result of human error. So non-sampling. So it doesn't actually have to do with how you sampled so much as human error. So we've talked about this before, computing errors, loaded questions, and things like that. And then non-random sampling errors. So non-random sampling error is the result of using a sampling method that is not random, such as convenience or voluntary response. We've talked about voluntary response in previous sections, and then convenience sampling is that quick and easy. We're not really caring about location or different strata or, keeping it nice and random so we can see what's really going on versus the population. And so lastly, go ahead and pause this, give these 3 a try, and then press play when you're ready to see the answers. And that concludes section 1.3.